Hello and welcome everyone. So today we'll be learning about a chromatin remodeling complex which is known as SWI-SNF complex. So when researchers were studying yeast mutants, the mutants which had reduced ability to grow in sucrose medium, while the mutants which had aberrant meeting switching type. So while studying those mutants, they found out a complex which was later named as SWI-SNF complex. So a few years later, they extracted this complex, and the role of this complex in chromatin remodeling was known. So the SWI in the SWI-SNF complex stands for switching mating types. Switching mating type, and SNF means sucrose non-fermenting mutants. So where the complex was found in switching mating type mutants and sucrose non-fermenting mutants, and that's why the complex is named after that. So the complex is known as SWI-SNF complex nowadays. So this SWI-SNF complex has a catalytic subunit. So it is made up of a lot of proteins, right? So it has a catalytic subunit which has ATPase activity, ATPase activity which hydrolyzes ATP, and that energy is required for chromatin remodeling. So it has a ATPase subunit which belongs to SNF2 family of the DNA dependent ATPases. So this SNF2 family of DNA dependent ATPases have different subfamilies. The SWI-SNF family, ATPase family, the imitation switch ATPases, which are also known as ISWI, the chromodomain and helicase-like domain ATPases, CHT. You remember when we were learning about nucleosomes, the nucleosomal assembly, we learned about a research paper which was published in 2005, and they used the CHD1, this chromatin remodeler, to establish the fact that both histone chaperones as well as chromatin remodelers alongside the ATP, they enhance the nucleosomal assembly. So I hope you remember the CHT1. So that is the chromodomain and helicase-like domain ATPase. Okay, and the other one is inositol requiring AT, iono AT. So these are the subfamilies of the catalytic domain of the chromatin remodeling complexes. Okay. So what was observed that there are two mating types in yeast, alpha and A. So when they have the yeast has to switch those mating types, it is possible due to HO endonuclease. This is only possible in the presence of HO endonuclease. So the gene encoding HO nuclease is in closed chromatin configuration. So then SWI-SNF complex works on this closed chromatin, and it converts it into the open chromatin configuration so that the gene encoding HO endonuclease is now accessible. So the polymerase can come and it can transcribe the gene, and HO endonuclease is formed. So when the yeast were mutated and the SWI-SNF complexes were mutated, it was unable to convert this closed chromatin structure to open chromatin configuration, and that's why it couldn't the gene couldn't encode the HO endonuclease, and that's why the aberrant switching type was observed. So the mechanism by which SWI-SNF complex works is different. So let's have a look at the mechanism first. So the possible mechanism which was suggested was something like this. So assume that this is a DNA, this is a DNA strand. Okay, so the SWI-SNF complex, this is what's happening. Just a second. Okay, so assume this is a DNA strand and this is SWI-SNF complex. And this DNA is occluded by nucleosomes. Reminder that this is not a normal DNA, this is a nucleosomal DNA. Okay. So this VSNF complex can they are recruited by some site specific or sequence specific binding proteins. We have already learned it in the previous videos. You can check that in the playlist. Okay, so those site sequence specific binding proteins guide the chromatin, this complex, to the DNA, and once this is recruited, what happens that it is recruited around 20 base pairs upstream the its own binding sequence. Okay, so when it is recruited over here, then the catalytic, the ATPase domain, the catalytic domain of this VSNF, it hydrolyzes ATP. Okay, and this creates the energy. So this energy is so due to the help of this energy, a DNA loop is formed. Okay, so a DNA loop is formed. So this is the DNA looping out of the nucleosome. Okay, and now the more you hydrolyze ATP, the longer the DNA loop becomes. So thus, due to the looping out of the DNA, now this region is accessible to the transcription factors uh, and the polymerases. Okay, so that this is how the SNF complex works. So the major consequence of this process is that when the when this DNA is looped out of the nucleosomal structure, what happens is that the linker DNA, the linker, okay, linker DNA, it get it gets short. Okay, so what happens, this is nucleosome. Okay, this is a histone core around which okay, this is the so this is the DNA. Okay, so this is the nucleosome core around which the DNA is wrapped. So what happens is, due to this SNF complex, this DNA loops out. Okay, so this DNA is now looped out. So due to the looping of this DNA, the linker DNA, the linker DNA becomes shortened. So imagine that you are pulling this thread from this direction. Okay, you are pulling it from here. So when you are pulling it, you're gonna form something like this. So due to this looping out of DNA, this becomes short. Okay, so this is the major consequence of SWI-SNF working. So this is how SWI-SNF work. And what does it do? So what does SWI-SNF do? It either disrupts the nucleosome, okay, for converting the closed chromatin configuration to open chromatin configuration. These are the possible mechanisms which SWI-SNF adapts. It either it disrupts nucleosome, or it increases the binding of transcription factors to nucleosomes. It can mobilize nucleosome on the same DNA stretch. It can transfer histone octamer to different DNA stretch. So due to the transfer of histone octamer, now the DNA stretch is available for the transcription. It can displace the H2A, H2B dimers. Again, the DNA histone interaction is disrupted non-covalently. Then it can generate the super helical torsion in DNA. It can bind preferentially to DNA and nucleosomes without DNA sequence specificity. This is a very important fact, that it can bind to the nucleosomes without the DNA sequence specificity. It can interact with transcription factors to localize the chromatin region whose transcription level is to be altered by nucleosome repositioning. So these are the all possible mechanisms that CSNF complex adapts for making the changing the chromatin configuration. So the protein complex VSNF has 12 subunits. Okay, as discussed, it was first discovered in yeast. So the catalytic subunit in the yeast is known as SWI-SNF. In the humans, it is known as BAF, while in Drosophila, it is known as BAP. Here, let me show you the diagram. So in the humans, it is known as BAF. The HBRM is the center of this BAF, the catalytic subdomain. So along with this catalytic subdomain, along with this ATPase, other proteins come together and it forms the holo complex which performs the chromatin function. So that's it for today. In the next class, we'll learn about the assays for chromatin remodeling. Till then, see you. Bye bye.